Hi, I am Ayda Başgül Martin. Welcome to my channel. If you like my video and watch more, please feel free to click the subscribe button if you haven't done so. And we will talk today the neuron anatomy and physiology. We will define the neuron and neuron features, describe the functions and location of these neurons, and identify the structures of a neuron on an anatomical model, as well as recognize the multipolar neuron on microscopic slides. How we do classification? We classify the neurons according to either their structure, their shape, or their function. According to their structure, you see there's a cell body and how many branches takes out from the cell body. If there is only one branches takes out from cell body, we call them pseudo unipolar neurons. They are generally found in spinal ganglia or another ganglias. And bipolar, they are sensory, by the way, they are sensory neurons. Bipolar neurons, bi meaning two, right? There is a two branches from the cell body. They are the sensory or neurons as well. They can be found in they specialize to take the taste, take the light in our eyes, or hearing in our inner ear. Multipolar neuron found in the central nervous system, and most of the neurons are multipolar neurons. What that means? They have more than two branches takes out from cell body. As you may see, there is plenty branches in multipolar neuron. When we look to functions, they can be either sensory, interneuron, or motor neuron. Sensory neurons get information about what's going on inside and outside of the body and bring that information into the central nervous system so it can be processed. And when you look at the interneurons, they're found only in the central nervous system, connect the one neuron to another, integrate incoming signals to determine whether or not the information should be passed along. Motor neurons got information from other neurons and convey the commands to the muscle or another neuron or glands. Okay, let's move on. When we look at this model, we will see the cell body. We will see some extension, some branches, and we call them dendrites. You will see the axon is a long single long branches, and axon forms from the cell body, and just before axon forms there is a triangle zone before the axon we call axon hillock. Here in this area summation happens. What that mean? Incoming signals can be either excitatory, which means they tend to make the neuron fire, generate an electrical impulse, or inhibitory, which means that they tend to keep the neuron from firing. And if an axon arises from the cell body at a specialist area called the axon hillock. Now, if there is 8 stimuli coming to the neuron, and 5 of them is excitatory and 3 of them is inhibitory, what will gonna happen? The summing up will happen in axon hillock. 5 minus 3, that means there is 2 excitatory neuron. That means that neuron will fire, will create the action potential because there's 2 neuron is telling to the excite. Okay, again you see in the right side all the organelles, smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi, mitochondria, and as you see the rough endoplasmic reticulum in the neurons specialized to form the Nissl bodies or chromatophilic granules. You can see on the left side nissil bodies clearly. It's like a granules. It's like a dots, right? A little bit bigger than dots on the neuron cell body. They may change if there is a harm to the neuron. They may change, and the shape and color change, and we use this in practice for diagnosis. Again, let's see another model. We see the dendritis, we see the synapse, axon terminal, axon hillock, axon itself. When we look to the left side, we see another cell that axon of the neuron covered the axon of the neuron. It's like a hot dog bun, right? We call this Schwann cell again. Between the Schwann cell, there is an opening. This opening we call node of Ranvier. Node of Ranvier. Schwann cell produce myelin, and that myelin sheet insulate the axon. So stimuli, electric stimuli, will not travel alongside the axon in myelinated neurons. However, it will jump from one Ranvier node to another one. We call this saltatory conduction, and it allows the action potential moves forward faster than unmyelinated neurons. Again, another model, we can see the 1 nucleus, 2 nucleolus, and 3 rough endoplasmic reticulum, 4 axon hillock, 5 axon, and 6, as you may see, dendritis, 7 terminal knobs, 8 Schwann cell, 9 neurolemma, 10 node of Ranvier, and 11 neuromuscular junction, because this neuron communicates with a skeletal muscle, and 12 motor end plate. And when you look at the histology under the microscope, you can see the motor neurons. I use the pointer to point out the motor neuron. How I know this is a motor neuron? Because all multipolar neurons are motor neurons. And you see there is more than 2 branches comes out from the cell body. And at the same time, you see some dots between these neurons. Those dots actually represent the satellite cells. So this is this slide taken from this specimen taken from perfect nervous system. Okay. Now, when we look to the terminal endings, this is a good model of the synaptic knob. And number 1 is representing axon. Number 2, synaptic bulb or synaptic knob. And number 3, showing the ion channel. 4 is the synaptic vesicle. Number 5, representing the neurotransmitter found in the synaptic vesicle. And number 6, as you may see, presynaptic membrane. Number 7 is a neurotubules, and number 8, this bluish one, is the branches of rough endoplasmic reticulum. Let's test your knowledge. Do you remember what was number 2? If you said axon, I am happy to see you learned it. Good. In the same way, make sure to learn all of the features. Thank you for watching. I hope you will like, share, and subscribe. That will motivate me to do more videos for you. Thank you.